Cell as a basic unit of life. Cells are the basic unit of all living things. Robert Hooke, in 1665, was the first to study and record cells by using a microscope. Robert Brown, in 1831, discovered the presence of nucleus in a cell. J.E. Purkinje, in 1839, gave the term protoplasm to the living contents of a cell that is surrounded by a plasma membrane. The entire content of a cell, comprising the nucleus and the cytoplasm, is known as protoplasm. In 1838, German botanist Matthias Jakob Schleiden found that all plants are composed of cells. Similarly, in 1839, Theodor Schwann proved that animals are also made up of cells. Rudolf Karl Virchow, a biomedical scientist, in 1855, was the first to recognize that all cells come from pre-existing cells by binary fission. So, these last three scientists put together the principles of cell theory, which are like this. All living things are made up of cells. Cells form basic unit of all living organisms. All cells come from pre-existing cells. The study of cell and its parts is called cytology. Advanced version of electron microscope has made study of the biological material very extensive and accurate. Cells are the basic unit of all living things. Cells are the smallest structural unit that can sustain all the required functions of life. All organisms are composed of cells. A cell has all the physical and chemical components needed for life. This is Spirogyra, a free-floating filamentous alga. Its cells are cylindrical in shape and are connected end to end. The cells of an onion skin are generally rectangular in shape with prominent nuclei. This is a monocot plant. Here are the epidermal cells of this plant. Let us compare the cells with the bricks of a wall. The brick is a part of the wall that forms building of different shapes. Similarly, cells arranged in some defined pattern form tissues, which in turn form different organs of a body. Bricks arranged in a systematic manner form a wall. In a similar fashion, identical cells join and form a structure known as tissue. Just compare the organization of small units in both these structures. There are some organisms which are composed of only one cell. These are known as unicellular organisms. They carry out all their functions within a single cell. Euglena is a single-celled microscopic alga. It is a unique organism that has chloroplasts and can make its own food. It is not completely autotrophic, as it can also absorb food from its environment. Euglena move by a flagellum, which is a long whip-like structure that acts like a little motor. Paramecium is a single-celled freshwater animal. It has tiny, hair-like things called cilia all around its body. Paramecium moves by beating the cilia back and forth, often changing its direction. Amoeba also has only one cell for its entire body. It moves by changing the shape of its body and forming pseudopodia. It also engulfs the food with the help of pseudopodia. There are two main types of cells: prokaryotic cell and eukaryotic cell. These two types of cells differ in many ways. Let us discuss some important differences between these two types of cells. Prokaryotic cell is the most primitive form of life found on the earth. It lacks a defined nucleus as well as other membrane-bound organelles. Its DNA is of floating kind and confined to a particular area in the cytoplasm, known as nucleoid region. So we can say that a prokaryotic cell has a very simple internal structure, as it does not contain membrane-bound organelles. Prokaryotes belong to kingdom Monera. Examples of prokaryotes are all bacteria, cyanobacteria, and purple bacteria. Eukaryotes are generally more advanced than prokaryotes. Characteristic features of eukaryotes are the presence of defined nucleus, numerous membrane-bound organelles, and complex internal structure. There are many unicellular organisms which are eukaryotes. Most protozoa are unicellular eukaryotic organisms. Example: paramecium, amoeba, dinoflagellates. Although they are unicellular but contain defined nucleus and membrane-bound other cell organelles. All multicellular organisms are eukaryotes with very well-developed cells. Like humans, dogs, trees, all these have different types of cells forming highly developed systems. Now let us do a small activity to prepare fresh mount of plant tissue. Objective of this activity is to study that living organisms are made up of cells. Procedure. For performing this activity, we need glass slide, cover slip, dropper, microscope, needle, onion, iodine solution, forceps. Put a drop of water on a clean glass slide. Cut a small section from the scale leaf of an onion. Use the forceps to peel off a small piece of epidermal layer from the scale leaf of onion. Keep it on the slide gently. To avoid air bubbles, use needle to lower a cover slip onto the epidermal layer. Look at the unstained slide under the microscope. Microscopic view of an onion skin shows several rectangular cells, each with a small spherical nucleus. Add a drop of iodine between the cover slip and slide to obtain a stained view of the epidermal layer. Now again look the epidermal layer through the microscope. Nucleus becomes slightly more prominent with iodine. The cells have definite shape. This is because of the presence of cell wall. Observation. When the slide is viewed through a microscope, rectangular-shaped cells of onion peel are visible. Do an activity with an objective to study that living organisms are made up of cells. In this activity, sample used will be of cheek cells. Procedure. For this activity, we need glass slide, cover slip, dropper, microscope, needle, toothpick, methylene blue solution. Put a drop of methylene blue solution on a clean glass slide. Gently scrape the side of your mouth with the blunt side of a toothpick. By doing so, you collect some of your cheek cells. Transfer the scraping onto the drop of methylene blue solution kept on the glass slide. Use a needle to lower the cover slip to avoid air bubbles. Set it under a microscope. Observe the cheek cells. The nucleus and cell membrane are clearly visible. These cells are irregular in shape, unlike the rectangular shape of the onion cells. Observation. When the slide is viewed through a microscope, irregular shaped cheek cells are visible.